Hello, everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, Interim Director of Creative Services for The Scientist, and I'll be moderating our discussion. Today, our speakers, Dr. O'Donnell Tormé and Dr. Huang, will be sharing their views on immunotherapy's role in the medicine of today and tomorrow. Is immunotherapy living up to its promise, and what will it take for us to realize its full benefits? We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during our Q&A session. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. Beckman Coulter develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. For over 80 years, our products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. Scientists use Beckman Coulter life science research instruments to study complex biological problems, including causes of disease and potential new therapies or drugs. From basic cellular analysis to advanced cell sorting methods and flow cytometry applications, count on Beckman Coulter for efficient and reliable solutions to today's research challenges. Scientists can also optimize their workflow and increase laboratory efficiency with our liquid handling, centrifugation, and custom integration solutions. Our sponsor has provided us with some helpful resources related to the study of immunotherapies, and we've posted these in our resource list located on the left side of the screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Jill O'Donnell Tormé. Dr. O'Donnell Tormé is the Chief Executive Officer and Director of Scientific Affairs of the Cancer Research Institute, a nonprofit organization founded in 1953 that is today the global leader in supporting and coordinating research aimed at harnessing the immune system's power to conquer all cancers. She joined the organization in 1987 and has been chief executive since 1993. Prior to joining CRI, she served as a research associate in the Department of Medicine at Cornell University Medical College and was a CRI postdoctoral fellow in the Laboratory of Cellular Physiology and Immunology at the Rockefeller University. Dr. O'Donnell Tormé serves on the boards of SFO, Science Foundation for Oncology in Zurich, Switzerland, the City University of New York, Richmond University Medical Center, and the Staten Island Foundation. In 2013, Dr. O'Donnell Tormé received the Cancer Research Institute's Frederick W. Ault Award for New Discoveries in Immunology, which the Institute awards to formerly funded postdoctoral fellows who make significant contributions to immunology research. Dr. O'Donnell Tormé.
Hi, thank you very much for that introduction, and I'm very pleased to be here. So I guess we'll get started. And the topic today is immunotherapy living up to its potential? And I hope by the end of the next few minutes I'll be able to assure you that I believe it is. So as many of you know, immunotherapy has now become the new darling of oncology. Science magazine named it the Breakthrough of the Year in 2013. It was ASCO's top advance in 2016, and at ASCO's recent meeting in June, it was certainly the topic of many talks throughout the meeting. Very big change from what it would, I'd say, 10 years ago when immunotherapy was kind of off to the side in a small conference room on the last day where no one attended. So we're clear that immunotherapy is certainly center stage when it comes to research and oncology. And besides being an area that's certainly of interest in the scientific and medical community, it has also reached into the popular press. There are many, many stories that have been released both in print and on radio and on TV, and probably most noteworthy is the story about Jimmy Carter's great response to one of the checkpoint blockades. So I think whenever this happens and the popular media picks up on successes of new cancer treatments, we're always worried about, is this an overly hyped story that gives hope to patients but gives too much hope without bringing it down to reality? And certainly, when it comes to the field of immunotherapy, this would certainly be a question that would arise very rapidly, because cancer immunotherapy over the years has certainly been plagued with a lot of overly hyped expectations, and then which were certainly then not come to fruition, and everyone lost a lot of optimism about the field in general, and then we would get these blips. So this is a slide I've put together, which is a little bit complicated, but in essence, what I'm trying to show is that since 1970 to the present, and on the y-axis, you're going to see sentiment here. But you can see that it was already back in 1970 when monoclonal antibodies were declared the magic bullet for cancer. And for those of you old enough, you remember seeing this on the cover of Time magazine. The next, I'd say almost 2 decades that followed that, though, led to a lot of disappointment. The entire immunosurveillance hypothesis was challenged in terms of people no longer believing that the immune system was really there surveying your body and destroying cancer. The first monoclonal antibody that was tested for lymphoma actually failed in the 1980s. And it really took until, I guess, into 1986 when we finally saw interferon gamma being approved as a treatment for melanoma. So again, we had this really high expectation in the 70s. Throughout the 80s, there was really a dip and almost a disbelief that the immune system could ever be used to effectively treat cancer. And then we started to get some positive responses, with interferon followed by BCG getting approved for bladder cancer in 1990. And then I put on here, I think an important point is in the 1991, the first tumor antigen was identified. And this gave hope that we could actually target the immune system specifically to cancer. Again, following that in 1992, IL-2 was approved for cancer, but then we went into a little bit of a tailspin over that, with the probably 2 things opposing each other. Over the next decade or so, we saw a lot of monoclonal antibodies being approved. The first in 1997, rituximab, and followed, I guess, through up until 2010, there were probably 9 more monoclonal antibodies that had been approved. But during this time, there was an awful lot of failures, mostly centered around vaccines. And you can see with the dips here in red in terms of throughout 2002, 3, 5, 2006, and all the way up to 2014 when the MAGE-A3 vaccine failed. There were, again, a lot of skepticism. And I think the problem with this is that popular press and also people that were not truly cancer immunologists, heard this, and especially practicing, I think, oncologists, hear these results and basically threw the baby out with the bathwater. And I think we can all know that the hardcore cancer immunologists always held on to the belief that we could understand how to use this, but many times some of these trials, especially when it came to cancer, were being put prematurely into the clinic without really understanding mechanistically how the immune system really worked. And I think the real renaissance that we're seeing now really can be traced back, obviously, to first 2010, when Provenge, which was the first active immunotherapy to be approved for prostate cancer, and then closely on its heels in 2011 when we saw that Yervoy was approved for metastatic melanoma. And again, this became the anti-CTLA-4 checkpoint blockade, and it was the first treatment for melanoma that ever had an impact on overall survival for late-stage melanoma. And since that period, we've seen between 2011 to the present, there's been 13 different immunotherapies that have received FDA approval for 12 different cancer types. And there's obviously still been some failures over that time period, again, with vaccines, with GVAX and Nuvax failing in 2016. But again, we're seeing that neoantigen vaccines are starting to come back, and I think we're going to see moving forward the combination of vaccines with some of these checkpoints will give additional, I think, positive and hope for people that immunotherapy is here to stay. And I think most recently, we just know last week, ODAC actually recommended the approval of CAR T-cells. So again, there's an awful lot of optimism right now, and I think we have had a checkered past, but I think we're on a very good trajectory moving forward. And now the reasons for this, you can see on the next slide, that why would cancer immunotherapy be considered the answer? And I think it goes, for multiple points, it really comes back to what the immune system has evolved to do and what it's capable to do. First and foremost, it's universal, and it could be effective for all types of cancers. It's powerful. We know that it can change cancer into a manageable disease, if not cure it. It's adaptable because the immune system is always ready to protect us against any unknown challenges that come in. So as cancers mutate, your immune system can adapt to that and still mount a defense against it. We have seen over the past few years, even in not only in animal models but in patients, the responses of immunotherapy can be very durable. And this also can trace back to the fact that the immune system has memory. And it's capitalizing on this innate ability is one of the hallmarks of what the immune system can do, and again, one of the reasons why it is such an appealing way to potentially treat cancer. Again, it's a systemic treatment, so it's something that treats the entire body. It's synergistic and can be combined not only with different types of immunotherapies, but also with standard of care in terms of radiation and chemotherapy or with targeted therapies. And again, it can be very targeted. If you can figure out exactly how the marker on a cancer cell that can be seen by the immune system, you can actually be able to target that response to go directly for the cancer and leave aside normal cells, which is obviously one of the great hopes that any cancer treatment could do. So what makes it so exciting right now? I've kind of dealt with it a little bit in the timeline I showed, but the reason that immunotherapy is so exciting to the medical community at large as well as to the general public is that there has been significant clinical responses seen with both checkpoint blockades and with CAR T-cells. These are increased survival in late-stage cancers that previously were intractable to all standard cancers. I think that has been the biggest and foremost thing that has happened. As I stated before, the responses that we're seeing seem to be quite durable. There is little, but there is sometimes resistance to this that we're seeing more and more as time goes on, and the fact that though it is applicable to many different tumor types. And I think putting those points together is why we're so excited. So now just to take a little step back in terms of how do checkpoint blockades work. So first, to giving a little science lesson here, if you look at the first drawing here on the upper left side of the screen, this is showing you how the T-cell becomes activated. So for a T-cell to become activated to be able to become an effector cell that can actually seek out and destroy a targeted cell as cancer, it requires two signals. The first is that the T-cell receptor on the T-cell needs to see the antigen or the marker that it identifies in context of the major histocompatibility antigen that's presented by something like a dendritic cell. But if that one signal happens just by itself, that does not lead to T-cell activation, but in fact leads to dormancy, and the T-cell actually becomes dormant and died. What you really need is a second signal, which is shown on the left side on the lower diagram. And there is a second signal, a marker that's called CD28 on the T cell, actually binds to a marker on the antigen presenting cell, the B7. And it's when these two signals happen simultaneously that you get T cell activation. But your immune system has evolved in such a way, though it's there to protect us from foreign invaders, it's also there to make sure that you do not develop autoimmunity and attack self. You always want the immune system to attack non-self. So the immune system has natural checks and balances. And one of these is something that's called CTLA-4. And we can call CTLA-4 the brake. And if on the right diagram, the upper right diagram, you can see that at the same time that the two signals are engaging and activating the T cell, the T cell starts to produce something called CTLA-4 on its surface that can actually outcompete binding to the B7 molecule. So it outcompetes CD28. And when CTLA-4 binds to B7 that actually sends a stop signal, and it stops the T cell from becoming activated, and it becomes dormant. Now, what checkpoint blockades have done is that an antibody can be created that binds to the brake, the CTLA-4 molecule. And when that antibody engages the CTLA-4, CTLA-4 can no longer bind to B7, so therefore the T cell stays in an activated state. So probably more simply stated, in the next diagram, it's really that an anti-CTLA-4, an antibody against CTLA-4, is actually taking the brakes off the immune system. And you can think of the immune system as a car, and that the first signal is the T cell receptor antigen MHC binding is very similar to putting the key into the ignition. Now, when you put the key into the ignition of the car and turn it on, but the car doesn't move. The car doesn't move until you put your foot on the gas. And the gas pedal, in terms of the immune system, is CD28 and B7. And then, of course, when you want to stop the car, you need to put your foot on the brake. And the brake is CTLA-4 and B7. So if you disrupt that CTLA-4 binding to B7, you would stay in the activated go state, and the car would keep running, and the T cell keeps working. So again, this was the first, the anti-CTLA-4, or known as Yervoy, was the first checkpoint blockade that was approved by the FDA for treatment of late-stage melanoma. And this is what really opened up the excitement in the field. But I think even more so, beyond anti-CTLA-4, there are other checkpoint blockades. The one that is most popular and I think developed the most excitement in the field is the PD-1-PDL1 axis. Again, another checkpoint blockade, but in this case, the cancer cell has actually co-opted this natural checks and balance system that's in your immune system. So tumor cells are actually producing something called PD-L1. And PD-L1 binds to a receptor on the T cell for PD-1. And what this happens is that by this binding, by the T cell co-opting this, it's able to shut off the immune system and allow the tumor cell to almost be cloaked and not be able to be seen by the immune system. And then we have understand and learned that if you can create antibodies that bind to either PD-L1 on the T cell or to PD-1 on the T cell, you actually can stop that interaction and allow the T cell to now see and destroy the tumor cell. So this is what has actually delivered, again, a lot of great clinical responses. And this is just a slide that summarizes what has happened in terms of immunotherapy FDA approvals since 2010. So back in 2011, Yervoy, which is the anti-CTLA-4 antibody, was first approved. Since then, all of this is showing all of immunotherapies that have been approved by the FDA, but in red are all the checkpoint blockades. So currently there are 6 different checkpoint blockades, one against anti-CTLA-4 and several against either against PD-1 or PD-L1. And these 6 checkpoints have been approved for 7 different cancer types, and not only in late-stage cancer, but in fact, some of these have been approved for frontline treatment of cancers such as melanoma, bladder, and non-small cell lung cancer. So this is showing you that this is certainly an exciting and active field in terms of cancer treatment. And you can also see there is a pretty indication of, in terms of different levels of overall response rates that happen with these checkpoint blockades. Now, this is a graph that is really showing overall response rates to either PD-1 or PD-L1 in various cancer types based on the amount of somatic mutations that they have, so the genetic load, the genetic mutational load of these tumors. But the main point, and though that's showing you that there is a correlation between those cancers that have higher mutation rates, such as melanoma, have a higher response rates to these checkpoint blockades, as high as like 40 to 45%, while some cancer cell types have really no responses to checkpoints, such as prostate or Ewingsarcoma, have not shown really any response rate to checkpoint blockades. And then you see a variability between 10 up to 40%. So this is, I think, impressive. These are impressive responses, but it's also telling you that there's only a subset of patients in various different cancer types that actually respond. So we have a long way to go to making, though there is all this excitement about what checkpoints could do, and especially in cancers that really were not responding to any other treatment, there's a long way to go to make these response rates higher for all different cancer types. The other main point as we bring back in terms of the durability of immunotherapies, and this is just a slide showing you with a Kaplan-Meier curve, which showing the proportion of patients alive over versus time, and you can see that these curves are flattening out. And this is showing you the longest really track record we have is obviously with the anti-CTLA-4 antibody, but we're seeing this also with anti-PD-1s, that it's this tail of the curve flattening out, that patients are surviving for longer and longer times. That, again, makes us very excited about the potential for these immunotherapies to stay around and become a backbone for the treatment of cancer. So the other, besides checkpoints, the other immunotherapy I think that has really delivered the most excitement is CAR T-cells. And these are chimeric antigen receptor T-cells. So these are T-cells that have been genetically engineered to actually put an antibody binding domain, basically putting an antibody on the surface of a T-cell and being able to target T-cells specifically to whatever you want with the antibody binding domain, but getting the effector responses of T-cells. And these have not yet been FDA approved, but the response rates are truly remarkable when you see in different types of leukemias. Most of these are all being targeted to a single target called CD19, which are found on B cells. But you're seeing response rates, that range depending on the type of leukemia we're talking about, 60 to 90, 100%. And because of this remarkable response rates that we're seeing, and though these do come with some severe side effects, but have sometimes now we've learned as these have been in the clinic to control these. But some of those side effects are certainly, adverse events are certainly significant. But as I said, back just last week on July 12th, ODAC actually unanimously recommended FDA approval for CAR T-cells. So the expectation is that the first CAR T-cell will be approved by the FDA in short form. So the goal of cancer immunotherapy is really to raise the tail of the curve so that more patients can benefit. This is a very simplistic graph that's showing you when you have, basically it's survival versus time, and when there's no therapy, you can see, survival is limited. Conventional therapy does work in certain cases. We're seeing with cancer immunotherapies as monotherapies, just an individual treatment, the individual type of immunotherapy, you're certainly seeing response rates rising and you're getting this tail of the curve. But the hope is that combinations, by combining these checkpoint blockades or combining CAR T-cells with other treatments or with themselves, we can raise the tail of the curve and get more patients that are responding for longer periods of time. And combinations make sense, and this is a complicated slide, but you can really understand that combinations make sense because to get an effective immune response against cancer takes a variety of steps. It's not just a one-step process. You have to generate cancer-specific T-cells in the first place, and this takes multiple steps. You need processing and presentation of the cancer antigen. You need the presence of pro-immunogenic signals to activate those cells, and then you need to prime and activate T-cells to get them to proliferate as effector T-cells. So once the T-cells have been primed and activated, these T-cells have to traffic, when you see in terms of number 4 on this little rounded graph, they need to traffic to the tumor bed. They then need to infiltrate actually into the tumor bed, and that's where they have to do their killing, but they're going into a very hostile environment around the tumor, and there's an awful lot of different immunosuppressions. And so at each one of these steps, there's various ways that you can affect these different processes, either in a positive way or a negative way, and I've just listed some of them around the edge of this slide. And you can see that I think the field was extremely lucky that the monotherapies of something like anti-CTLA-4 or the first anti-PD-1 actually worked to such a significant effect, and that you were getting 20% response rates, and in some cases 30 or 40% response rates. Because I think if we had not seen that type of response rates, this entire field would not be where it is right now. But we're lucky that that percentage of patients could respond with a single antibody against either CTLA-4 or PD-1 or PD-L1, because you see that's really acting only at one of these 6 steps of this process. CTLA-4 really works over at step 3. The PD-1s are working around at step 6 in the tumor microenvironment. But in those patients that are not responding, they do not have all the rest of these steps working in conjunction with each other. And this is the challenge we face as the field. How can we figure out how do we get this active immune response? We have the proof of principle. When the immune response is triggered in the appropriate way, a significant portion of patients can respond in a very favorable way. But how can we figure out now how to convert those non-responders to responders? And I think that's where the future is, and that's where we need to do an awful lot of research. We are really at the tip of the iceberg when it comes to the potential of cancer immunotherapies. And I think the major goal of research that's happening right now is really turning non-responders into responders. Having been in this field for a very long time, we are really at a unique position. We've never before had such a large number of patients that have positively responded to immunotherapy. Unfortunately, we also have a lot of patients that have not responded. But this gives us a platform where we can ask questions from a research perspective that we've never been able to before. We can now interrogate different things. And I think we're seeing that you can kind of segment patients into 3 big buckets. There are those on the left side where their tumor looks like an immune desert. There's only tumor. This is on the bottom here. You can see is the tumor cell where the heavy line is, that kind of outline. So below that line is tumor cell. And you can see there are no T cells there. That's only tumor cells. The T cells are labeled in kind of the reddish-brown color. So this is an immune desert. And this would tell you that there's obviously, in patients that present this way, there's a T cell priming defect. So therefore, we have to get them to respond to the current available immunotherapies, we need to induce inflammation. And there's various ways you can do this, with vaccines, with cytokines, potentially even with chemotherapy. And so there's all of these potentials that we need to look in and to see what would work right in that patient with that presentation. Then we also, if you move to the middle panel, there's something called, this is kind of immune excluded. This is, again, showing that the tumor cell on the lower right-hand side of the picture, but above it, you can see there are T cells there that are labeled in brown, but they're not penetrating into the tumor bed. So this is a homing defect. The immune cells are being excluded from the tumor, and in this case, to get them to respond to immunotherapies, we probably need to find ways to overcome the physical and cellular barriers of T cells tracking into the tumor. And this is looking at things of like, how do we get rid of stroma? Or is there extracellular matrix that needs to be broken down? Are there other cell types that are involved here, like myeloid-derived suppressor cells or B cells that need to be looked at and inhibited, or are they inhibiting the T cells from getting in? And then there's the third type of presentation of a tumor, and this would be called an inflamed tumor. Here you can see there's obviously T cells that have actually gotten into the tumor bed, but these patients aren't responding to the checkpoint blockades. And this is because the T cells are being suppressed or inhibited at the tumor site. And this is where checkpoints would come in, or this is where targeted therapies could come in. And I think we have to understand, in terms of looking at this, from the patients that are not responding, and understanding what do we have to add to a cocktail of combination treatments that could deliver the response that we want to get an effective immune response. So to realize the maximum effectiveness of immunotherapy, my feeling, and of course the feeling of the Cancer Research Institute, is that we need more research and we need more collaboration and innovation. And I'm proud to be able to say that I feel the Cancer Research Institute continually focuses on this. We have been obviously since 1953 with a singular mission of focusing on supporting research that would teach us how the immune system functions and how it can be used as a treatment for cancer. And we do this, and I think a big question now that we have clinical successes is should we be doing only clinical research? And I think the resounding response to that from our side is that no, we need to still learn so much about the biology of the immune system. So we still need to do basic research, we need to do translational research, and we still need to do, obviously, clinical trials. And CRI has programs in all of these. You'll be hearing a little later from Alex, who is actually a recipient. He was a CRI fellow and a CRI investigator. So we're happy to see that the people we support go on to do great things, and you'll be hearing from them. There's also a need, I think, for more collaboration and innovation. And I think you see this happening in the ecosystem that is biomedical research right now, and especially that focus on immunotherapy. I think everyone's coming to realize no one can do it alone. And we need to have a collaborative environment where the academics work with industry partners, work with regulators, and of course, include patients in this entire mix. And so things have to be done a little bit differently than they've done before. And again, I think CRI has been innovative in this and being on top of it. We have our clinical strategies called our CRI Clinical Accelerator, and this is a really an incubator where we actually partner academics with industry partners in a space that really, I think, a not-for-profit can allow industry to have new shots on goal, to use the academic investigators to come up with the best ideas of how to do it, and to have an organization like CRI be able to broker access to various investigational agents that are owned by biopharma and have them used together in a clinical trial under our auspices. We also have recently partnered with the Parker Institute for Cancer Immunotherapy on a program called Tcells, which is the Tumor Neoantigen Selection Alliance. As I said very early on in the field, I think cancer vaccines, though they've gone through a very rough time in the past and really didn't produce many clinical responses, the field does believe that as these neoantigens, antigens that are being developed because of mutations that are happening in the cancer that are very singular to any given cancer patient, so that there isn't just one looking for a cancer antigen that's a universal cancer antigen across all cancers, even within one type of tissue type, but rather being able to find the neoantigens, those new antigens that are actually being produced in a given patient, and creating personalized vaccines to target the immune system to recognize that. And so we have partnered with the Parker Institute to create Tcells, which really has 30 organizations, both companies and academics, we're basically everyone having different algorithms on how to predict these neoantigens. And so this is actually bringing 30 people together to collectively test their different algorithms with the same tissue samples, the same samples from either from cancer and normal cells, to look for these genetic sequences. And then these predictions will be validated to see which are the most likely to be recognized actually by T cells. And with this way, we hope to be able to inform the people doing it so that we share this information so they can improve their algorithms, and we can find the right algorithms to use to be able to move forward into the clinic to identify what these neoantigens are so that we can create vaccines. We also have our Cancer Immunotherapy Consortium, which is a think tank of key players from the biomedical industry in terms of trying to see if there are issues that under a not-for-profits umbrella, we can actually have them collaborate in a way that they couldn't do on their own to answer questions by either combining data or working together to move the field forward in terms of addressing late-stage issues that are impacting getting more immunotherapies across the goal line and FDA approved. Obviously, for all that we do, we rely on donors to really support everything we do, and you can certainly learn more about the Cancer Research Institute at cancerresearch.org. So coming into concluding, I think that it's important to see what are the trending considerations in immuno-oncology. As I said, there's an awful lot more research that needs to be done, and I think there's certain areas that we are focusing on at CRI, and we see that these is what the applications we get in from scientists worldwide are trending in these areas. Obviously, a lot of research on the tumor microenvironment, because this is where the action is. This is where the T cells have to end up killing cancer cells, so we need to understand all of the players and cell types and infrastructure of the tumor microenvironment. The microbiome is another very hot area of research. We've come to see that commensal bacteria does influence the immune response and also can influence the response to things like checkpoint blockade, so we need to understand better that interaction. Metabolism is also a very important area because it may predict T-cell function and patient response to immunotherapies. We've already talked about neoantigens. There's obviously many more checkpoints that have been identified besides just CTLA-4 and PD-1 and PD-L1, and these are all being investigated, and we need to understand how these can be combined together and how their mechanisms and pathways of actions can be synergistic with each other. Immune profiling, I think, is very important, really selecting which patients will respond to what treatments. And again, this will all be fueled by new technologies. But I think, the future is that immunotherapy is living up to its promise, but clearly we're only at the beginning. We don't want to overstate what's going on. We have a proof of concept in patients, in a large majority of patients, in many different types of cancers, that immunotherapy can deliver really effective responses that are durable and in some cases potentially curable. So we've had this big leap from people not believing that the immune system could ever be used as a treatment for cancer to having kind of a very big bandwagon that everybody is jumping on, saying that immunotherapy is revolutionizing cancer treatment, which I believe it is. But the next big jump is probably not around the corner. I think we're going to see very steady progress as more research delivers, as we understand mechanistically what's happening, as we can intelligently combine different things together to bring to the clinic. But failures will happen, and I think that's one thing we should be clear of, especially as more and more of the popular press talks about immunotherapy, and they'll be talking about failures that occur. And I think we want to, I think we're not in those little blips that we were before, that one failure could kill the whole field and kill the enthusiasm around it. But I think we have to be realistic about expectations. But I do believe the future is that immunotherapy will be a backbone of cancer treatment, and that the future is really about precision immunotherapy. We need to understand how, at the time of diagnosis, we can find out what a given patient needs. What combination do they need of drugs to bring together to get them to mount an effective immune response. And it's going to be personalized. So it's precision immunotherapy that's personalized, and we need research that will be able to help us identify what treatment is right for which patient. And I think that's what the future holds. So thank you very much. It was a pleasure to speak with you.
Thank you, Dr. O'Donnell-Tormé. As a reminder to our audience, you may ask questions at any time using the question box. The speakers will have an opportunity to respond after our next speaker. And that next speaker is Alex Huang. Dr. Huang is a professor of pediatrics, pathology, biomedical engineering, and general medical sciences at Case Western Reserve University. He holds the Klein Family Foundation Chair in Pediatric Oncology at Rainbow Babies and Children's Hospital's Angie Fowler Adolescent and Young Adult Cancer Institute. Currently, he leads the Tumor Immunology and Immunotherapy Interest Group and serves as co-leader of the Hematopoiesis and Immune Cell Biology program at the Case Comprehensive Cancer Center. In this capacity, Dr. Huang oversees immune cell-based and immune modulatory cancer therapeutic development. Most recently, Dr. Huang served as a member of the Cancer Moonshot Initiative Blue Ribbon Panel Working Group on Cancer Immunotherapy, as well as the inaugural Fellow of the Harrington Discovery Institute. Dr. Huang's research focuses on understanding aspects of immune regulation at the tissue level in cancer, autoimmunity, and trauma. The ultimate goal of his research program is to utilize the power of the immune system to control aggressive and metastatic cancers in pediatric, adolescent, and young adult patients. Dr. Huang?
Thank you so much. I hope you can all hear me. Thanks to Jill for laying out a great historical perspective as well as current status of where we are in immunotherapy. And I will continue on that trend of thought, but I thought I would take advantage of the fact that I'm a pediatric oncologist, and up to this point, most of the discussions we talked about immunotherapeutic approaches have been focused on many of the tumors that affect the adult population and not necessarily adolescent young adults. So I would take the first few slides to sort of lay the groundwork of where we are in a treatment of patients, adolescent, young, and children with cancer, and then I'll lay out some of the additional challenges facing this population, and then maybe lay out some of the future directions and using it as an example of how basic science research will continue, hopefully, to fuel the next generation of immunotherapeutic approaches. And so nothing to disclose as far as financial relationships. So the facts are that for pediatric cancer, believe it or not, it is the leading cause of disease-related death in children and adolescents in the United States. We have actually, as a community of pediatric oncologists over the last 5 decades, have seen incredible progress in providing cures for children and adolescents who are suffering from cancer. And this is largely through a very collaborative approach in the pediatric oncology arena to treat patients according to protocols across the United States so that we can actually collect a large data set for a patient whose disease incidence is relatively rare. Now, how rare is this? Well, here's a table showing all the causes of death among children and adolescents up to age of 19 as of 2013 in the United States. As you can see, among patients between 1 to 14 years of age, malignant neoplasms actually rank number 2 behind accidents. And for teenagers, it ranks number 4 behind accidents, intentional self-harm, and homicide. So taken together, cancer as a disease category actually is predominant in children's population. And as a number, we probably estimate around 15,000 cases of children and adolescents diagnosed with cancer in the United States annually. So it is a significant problem, and it's obviously the leading causes of death among children and adolescents for disease perspective. Now, over the last 5 decades or so in pediatric oncology field, we have actually seen an incredible progress in terms of the patients who are able to survive at least 5 years and beyond from their initial diagnosis. The graph on the left showing a variety of different common pediatric diagnoses of cancer, and in gray bar showing what are the survival rates in the 1960s. So if you are a child diagnosed with acute lymphoblastic leukemia, your chance of survival is below 10%, whereas in present time, up to 2007, you have 95% chances of surviving past 5 years. And we've seen that across the spectrum of diseases. On the right upper graph shows an overall survival of 84%. And on the bottom right, showing that we have continually improved the overall survival of children and adolescents with cancer. And in fact, this improved outcome for childhood cancer therapy using combination of surgery, radiation, and combination chemotherapy has really been championed as one of the success stories in oncologic treatment. Although we have largely as a field been a victim of our own success, in that there are problems associated continually with these kinds of therapeutic outcomes. For example, the improved children outcome comes with a very heavy price for survivors. And Joe mentioned earlier about the plateau effect. For adults, if you have additional 2 or 3 decades of life after a certain therapy, that is tremendously great, obviously. Now, for a child who is diagnosed with cancer and we're able to provide a cure within the first 5 years or so, this child hopefully will go on to live for another 5, 6, 7, 8 decades. But we're seeing now in practice is that children who are cured of their cancer with very heavy chemotherapeutic regimens are coming back with a lot of long-term side effects. The second thing is that we still are not doing very well in terms of refractory recurrent or metastatic disease. That continues to be a big challenge despite very intensification of chemotherapy. And then the third problem is that there's a specific population, adolescents and young adult population, that really have not benefited from all the advances in traditional chemotherapeutic and surgery radiation approaches. This graph shows that patients who survive their diagnosis of cancer beyond 5 years have a disproportional higher rates of mortality due to a variety of reasons. And they actually, over the last ensuing 2 or 3 decades has a 20% drop in their overall survival compared to their normal counterparts. And it just speaks to the point that, we need to continue to seek out in potential new therapeutic options that will lessen the toxicity, which is one of the primary goals in many of the ongoing clinical trials in pediatric cancer. And I think this is where immunotherapy can come and play a very important role in the years to come. The second challenge I mentioned was that in diseases such as Ewingsarcoma or other childhood sarcomas, osteosarcoma, and others, that if you are a patient diagnosed with metastatic disease, there is the outcome is continue to be very, very dismal. So the graph on the left showing whether you have a Ewingsarcoma extraskeletal in green or skeletal in blue, if you're diagnosed without any metastatic disease, your overall survival is around 80%. If you are a patient presenting with a metastatic disease at diagnosis, your overall survival then drops dramatically down to below 20%. And this despite the fact that we, over the last 2 decades or so, has increased our intensification in terms of durations and frequency of chemotherapeutic combinations, and we really have not seen any improvement in that. So we're hitting a plateau on what we can do with traditional chemotherapeutic approaches to treat cancers such as sarcomas, especially in metastatic disease. And I think, again, this is where immunotherapy potentially can be a very important adjunct. The third population I mentioned is the adolescent young adult population. The graph here shows the continual improvement in patients in the first decade and a half of patients who are diagnosed with cancer. They continue to show an increase in their overall survival, and this is mirrored by patients in the latter decades of life in all kinds of cancers. It's surprising to find out that for patients who are in this middle age, 15 to 30 or 40 age range, they really have not benefited much from all the advances that we as an oncology community have championed so much. And this obviously is due to a variety of reasons, which I don't have time to talk about. But there is an unmet need for patients in this particular age group. And for that, NCI and other foundations have really poured resources now into addressing this unmet need for the patient populations that happen in this age group. So the survival gap is something that I think in the future coming years, immunotherapy can contribute to that. Now, Jill has already outlined a lot of the promises of immunotherapy, so I'm going to quickly go over these. Obviously, immunotherapy offers the opportunity for effective immune cell function that can traffic to places where potentially other small molecules cannot get to. They rely on cell-cell interactions in a very cell-specific manner. And over the past several decades, there are a variety of approaches trying to activate the immune system to fight cancer with differences in overall outcome. But ultimately, the promise is that our immune system can be exploited to attack cancer with specificity and sensitivity and ultimately memory, as Jill mentioned. The fact is that cancer develops despite evidence that there are specific anti-tumor immune responses. They're capable of eliminating the malignant cells in patients' body. And I think there are a variety of background data now showing that this is in fact true. However, patients do develop cancer, and in those cases, there are many components of the immune system that are defective in patients that presents with cancer. But we now know that the immune system, when properly activated in the right context, can be highly effective against therapy-refractory cancers. And Jill outlined the successes we've seen with checkpoint blockades. And as she mentioned, we're only scratching the surface of how we can utilize immune responses to treat a variety of so far therapy-resistant cancer types. And this is just another outline to show that there have been several approvals for immune-based therapeutics, and this list will continue to grow at a steady pace. And I've been fortunate enough to be affiliated with Cancer Research Institute, who continually fueled all the basic science translational research in terms of using the immune system to fight cancer. And this just outlines some of the notable people that I've been fortunate to affiliate with, and showing on the lower right-hand corner the amount of enthusiasm, not just from academia, but obviously from funding private industry into the immunotherapeutic arena. And so I think we are really in a very exciting environment, and my hope is that some of these things will trickle down also to benefit pediatric and adolescent young adult patient populations. Now, just a few sort of general comments about immunotherapy, and then I'll talk about a specific example of where we think we're going to go in the future. Now, obviously, in terms of immunotherapy, I think one of the approaches that many people are familiar with is bone marrow transplantation, the hematopoietic stem cell transplantation. In fact, that's one of the earlier form of immunotherapy that's been shown to be very effective. We know that the graft T cells from donors can react against recipient tumor cells in a process called graft versus leukemia effect. But the same mechanism of killing or related mechanisms of killing can also greatly affect the recipient's normal tissue in causing problems such as graft versus host disease. And this is all due to the fact that immune systems are so sensitive and specific for individual that when they are transplanted into another foreign environment, their activation status can be changed. Now, just for the audience, and Jill has already mentioned several approaches, including checkpoint blockades and CAR T cells. What I thought I would do in the next few slides is to quickly summarize a few strategies that sort of hopefully summarize a lot of what's going on right now in the field in terms of immunotherapy. So one of the strategy one of how immunotherapy can be applied is to manipulate and expand immune cells that we think can be tumor specific outside of the body. And this can be exemplified by the approaches taken where we isolate T cells from patients' either blood or their draining lymph node or the tumor cell tissue itself, and isolate T cells to then expand them ex vivo in a petri dish, whether to expand them in the phase of growth factors, or as Jill mentioned about chimeric antigen receptor T cells, to manipulate them genetically with constructs that will target these T cells to specific sites against a specific tumor antigens. And then we expand them to large numbers, infuse them back into patient, and hopefully the cells will then seek out relevant cells to target them. And this can be done either on T cells or more recently on natural killer cells. Similar thing can be applied as well. So there's one of the approaches, and obviously we've seen success stories, and this is actually one of the areas where pediatric oncology has led the way in terms of the application of CAR T cells for treatment of B-cell leukemia by targeting specific antigen CD19 and now additional targets on leukemic cells. And so this has really led to a lot of enthusiasm in the CAR T cell area, but we continue obviously to develop in this arena by targeting other additional targets such as CD22 and other things. Now, there's been some effort in developing CAR T cells against solid tumors, not all easily done, and part of it is due to the struggles with identifying antigens. They are truly tumor specific without causing a lot of toxicities by targeting the CAR T cells against normal tissues. There are now additional CAR T cells, for example, against HER2 antigens for advanced sarcomas. The challenges are that they're suitable targets beyond B-cell malignancy, whether they're truly targets that we can find. The ability of patients' own T cell that can be manipulated and harvested. Sometimes if you have a very heavily treated patient, they do not have viable T cells to harvest. Such protocol sometimes becomes very difficult. And up to this point, there's some limited accessibility for patients and families to receive therapy, only in selected institutions, although this is rapidly changing as well. So that's first strategy. The second strategy is a direct activation immune system inside the body. And for this, Jill mentioned about some ups and downs in the past several decades with either tumor cells as a vaccine or using dendritic cells as professional antigen-presenting cells that pulse with tumor lysates or transfected with genes containing tumor antigens, using direct DNA vaccinations or nanoparticles in more recent developments. And the whole idea was to immunize patients with these vaccines and then use the patient's own body as incubator, and hopefully will expand the relevant T cell population that will traffic and kill the residual tumors in the patient's body. The third, obviously there continue to be applications of these and developments such as oncolytic viral vaccines for brain tumors, and dendritic cell vaccines in gliomas and other diseases, peptide vaccines for DIPG and other things. And I'm just mentioning here specifically in the pediatric oncology arena. Obviously, there's a lot going on in the adult oncology as well. The third strategy is something that I think very many people are familiar with. That is to use antibody to target proteins that are on the surface of tumor and not on normal tissues. They're represented here by these red triangles. And so there have been several development in this arena. And in pediatric oncology field, there's recently a anti-GD2 monoclonal antibody that targets on neuroblastoma has been approved by FDA, and they continue to have other development in this arena as well as a way of another form of immunotherapy. The fourth strategy is a large topic, which Jill also alluded to earlier in her talk, and that is the tumor microenvironment. Knowing that for some tumors, actually the majority that constitutes the tumor mass is not tumor cells themselves, but many components of the stroma, angiogenic cells, as well as immune cells, and how each of these components play a role in developing the tumor and how targeting each one of them might potentially benefit the ultimate outcome is a large area of investigation, and we'll stay tuned for that. And then the last strategy is that's already been talked about, which is the immune checkpoint blockade. And you know, Jill used the illustration of a car with a brake, and the blockade allows the brake to be off, allowing the T cell to be effectively killing the tumor. I used another illustration, and that is that when immune cells come in trying to attack the cancer cells on the left here, the cancer cell put up a shield to prevent themselves from being killed. And the shield comes in the form of the immune checkpoint blockade as one form of the shield. And for Star Trek enthusiasts, you'll remember that each of the Death Star has a force shield that protects them from attack. And but when the Alliance actually break down the shield generator, the whole Death Star can be blown up with one single shot. So I think over the last decade, we have found out that one of the weakness of the shield is this checkpoint blockade inhibitor, and PD-1, PD-L1 interactions. And this has already been talked about, where the engagement of PD-1 on T cells with PD-L1 on tumor cells allows the tumor cells to thrive and the T cell function to be diminished. And by blocking it, you allow the T cell to be supercharged and causing tumor death in the process. Now, the problem, and this is just another update in where pediatric cancer is in terms of immune checkpoint blockade application. We are now through the consortium testing some of the compounds now in relapsed pediatric solid tumors as well as other cancers, and so we'll stay tuned in seeing what the outcome will be. I think this will be a very exciting development in pediatric oncology. So the targeting a PD-1, PD-L1 can be effective in a fraction of patients. It's been mentioned already that in patients upwards of 45% can respond, but not all tumor will respond to therapy, and the therapeutic responses may not always correlate with whether the tumor expresses PD-L1 or not. And then the second thing to keep in mind is that PD-L1 expression on tumor cells, is not restricted to only tumor cells, but they can be expressed on immune cells as well. And the second thing is that PD-L1 expression is not static, and they respond to a variety of immune pressure. For example, gamma interferon is known to upregulate PD-L1. And so whether or not using PD-L1 expression in tumor specimen as the biomarker, it remains to be a controversial topic. And this is not a simple issue because for a variety of pediatric cancers, on immunologic specimen inspections, the PD-L1 expression can be variable. And so it led to some skepticism whether checkpoint blockade will actually work in pediatric cancer. And the skepticism is even bigger when considering the mutational landscape of pediatric cancer. And Joe mentioned this earlier, that melanoma and other adult cancers on the right side of this panel has higher mutational load that corresponds to a higher number of patients responding to immunotherapy. But on the left-hand side of this graph where you saw rhabdoid tumor, Ewingsarcoma, medulloblastoma, many of the pediatric cancer types belong to a low mutational burden tumors. And so it remains to be seen whether the trend holds where we saw in adult cancers will also be applied to pediatric cancers. So there are relatively low somatic mutations in pediatric cancers, and whether or not this will translate to the spectrum of peptide antigens and how patients will respond to the checkpoint blockade is yet to be seen. Now, the last few minutes, I'll just talk about some of the newer approaches on the horizon beyond PD-1, PD-L1. Where are we now? And offer some suggestions as an example of what can be done to not only to enhance the effectiveness of the PD-1, PD-L1, or CTLA-4, for that matter, approaches, but also to mitigate some of the side effects we see with severe autoimmune diseases in patients that actually respond to immune checkpoint. So I mentioned about this defense shield that the PD-L1 tumor put up to help themselves, and we'll just talk about one of the ongoing studies in the laboratory that was published last year to actually examine how tumor cells themselves regulate the expression of PD-L1. And the idea was that if we can actually target PD-L1 expression specifically on tumor cells, then there's a potential that we can bypass some of the systemic toxicities associated with the systemic administration of PD-L1 or anti-PD-L1 or anti-PD-1 antibody, and therefore potentially mitigate some of the autoimmune diseases that we see in patients. And this brings me to a topic, an enzyme called cyclin-dependent kinase 5, which is a serine-threonine kinase. It's been implicated in neurodegeneration. It's essential in CNS development. It has a role in myogenesis, vesicular transport. It's been shown that there are aberrant activation of CDK5 in multiple cancers, including prostate, pancreatic, and breast. There have been non-selective CDK inhibitors that hit the CDK5, but they are currently in phase 1 and 2 clinical trials for anti-tumor effects. Our laboratory, in collaboration with our colleagues here on campus, have demonstrated that the same enzyme actually plays a very important role in T-cell biology. And so it's an enzyme that has a double-edged sword in that if we effectively target tumors, it might have a positive effect, but if we're not careful, it might have a deleterious effect on T cells. So the overall, in long term, how this is going to apply in immunotherapy remains to be seen. But again, as I mentioned, CDK5 is important T cell biology and graft versus host disease. We've demonstrated that actually the same enzyme is important in IO2 transcription in T cells upon TCR engagement. That it modulates the IO2 transcriptional repressor, MSIN3A HDAC1, and in the absence of CDK5, the repressor complex remain on the promoter for IO2, and it limits the IO2 transcription for T-cell after T-cell activation. We've shown that T cells, they are diminished in CDK5 activity, actually has a diminished graft versus host disease in a fully mismatched bone marrow transplantation model shown on the graph on the right, on the lower bottom here. It's a very busy slide, but in panel B shows that the CDK5 deficient alloreactive T cells actually cause a minimal GVHD, and even though the GVHD effects diminished, in panel E here, it somehow maintains a GVL effect. So it's a target that affects how the cytokine production of T cells following activation, but somehow does not affect some of the membrane, surface membrane directed killing of the T cells against tumor cells. and potentially a potential target in this case, graft versus host disease, to mitigate a cytokine-related toxicity by maintaining some of the ongoing graft versus leukemia effect. And the story, just for the next few slides to talk real quick about where CDK5 potentially can play a role in cancer cells. Showing here is some of the outcomes data looking at tumors in glioma, melanoma, adenocarcinoma, and breast cancer, showing that tumors that express high levels of CDK5, shown in red curves here, universally have a worse outcome compared to patients' samples that have a lower CDK5 activity. And this is clinically shown also in a very common pediatric brain tumor, medulloblastoma here, the panel on the left showing that as you look at the level of CDK5 staining from left to right, there seems to be an indirect inverse correlation between CDK5 activity and the level of CD3 T cell infiltration. And so somehow the level of CDK5 expression in cancer cells is affecting how the host immune response is in the tumor microenvironment. We went on to show that in a syngeneic mouse model on the upper left-hand corner here, that the wild-type medulloblastoma, when they're inoculated into mice, causes a very rapid decline of the mouse. They also succumb to tumor, as exemplified by the black diamond, whereas the mouse tumors that are lacking CDK5 in orange actually has about 50% elimination of the tumor given the same dose. And on the middle right lower panel here, showing that these tumors that are eliminated, the mouse eliminated tumor actually develop long-term protection in that when they're rechallenged with the wild-type cancer cells, about 80% to 70% of these mice are able to now eliminate them. And this elimination is dependent on CD4 T cells, in that when mouse are depleted of CD4 T cells, you lose that protection. So exactly what's going on in the tumor microenvironment when CDK5 is present or absent, what we saw was that in the absence of this CDK5 enzyme, the immune cells has very high levels of PD-L1, as shown here in both panel E and panel G, both in the lymphoid compartment as well as in myeloid compartment, and that correlates with the amount of gamma interferon in the microenvironment, in that the interferon gamma is very high in tumors that lack the CDK5 activity. And what we examine in the tumor cells themselves, what's going on with the level of PD-L1 expression, we found that tumor cells that are lacking CDK5, exemplified here in the Western blot at the bottom, that they has a deficiency of PD-L1 expression, and that expression is correlated with a molecular regulatory change that encodes both the IRF1, IRF2, and IRF2BP2, in that the tumor cells, in the absence of CDK5, are unable to upregulate PD-L1 when it responds to interferon gamma. So in turn, making the tumor cells more susceptible to incoming immune attack, and they're unable to mount the defense shield as a result. And we have gone on to actually work on individual phosphorylation events and showing that there are specific phosphorylation that are affected by the presence of CDK5. And so we are now working on how these individual mutations or phosphorylation differences on the IRF2BP2 is affecting the regulation of PD-L1, in hopes that we can identify targets and other agents potentially that can affect differentially the amount of PD-L1 expression on tumor cells, and making them more sensitive to immune attack. So what we have now worked on is that this same regulation of how CDK5 regulates PD-L1 in response to interferon gamma axis can be applied not just in medulloblastoma, as we've shown so far, but also in other pediatric and adolescent tumors such as rhabdomyosarcoma. and we are in discussion, obviously, with companies that make inhibitors of the CDK5 to see whether or not we can use the agents to condition the tumor microenvironment so to make them more susceptible to immune responses and subsequent immunotherapy, whether it might be T cells or immune checkpoints. So the paradigm, as we already seen this graph before, is that the defense shield put up by cancer cells in terms of PD-L1 regulation allows the tumors to thrive and downregulate effectiveness of the T cells. The checkpoint blockades allows the T cells to be supercharged and causing the death of tumor cells. And what we're hoping to now continue to identify is other agents that allow similar event to happen by removing PD-L1 expression or diminish PD-L1 expression on tumor cells and allowing the T cells to kill tumors, and obviously finding ways to diminish the effect of the anti-PD-1, PD-L1 toxicity on systemic administration into systemic tissues. So just as an example of what is to come, and I think these efforts in basic science as well as other ongoing clinical trials and different combination therapies to take advantage of immune checkpoints in face of vaccination strategies, or how do we strategize patients so that we can either treat patients first with multimodal chemotherapy followed by immunotherapy, and how to preserve immune function in face of very aggressive chemotherapeutic agents. I think these are all challenges that we need to figure out as a field. So there's been major strides achieved in pediatric cancer and obviously in adult cancers as well. There major challenges remain, as I already outlined. It's important alternative therapy, and I think in some cases, at least in adults, it's become frontline therapy for immunotherapy. I think pediatrics, again, as I said, we're victims of our own success. I think the challenge will be how do we move immunotherapy, how do we identify opportunities to move immunotherapy into frontline in certain cases of pediatric cancer. That remains to be seen. We have seen CAR T-cell therapy successes, antibody therapy, and hematopoietic stem cell transplantations. Obviously, it's been very successful in the past. And the ongoing development in oncolytic viruses and other immune approaches, including checkpoint blockades, will be a very welcoming change in pediatric oncology. So thank you for the opportunity, and I think Jill and I will be taking some questions.
That's right. Thank you, Dr. Huang. The audience has submitted several questions, so let's get to them. Dr. O'Donnell-Tormé, what are the primary differences between immunotherapeutic strategies for solid versus hematologic cancers?
Well, I mean, Alex may be better able to answer this question, but I think what we're seeing is that the hematological cancers, which are certainly very responsive to the CAR T cells, it's because of the fact that these have mostly been focused on B-cell leukemias, and there is a single target, CD19, that's only on B-cells. So you can target this immune by doing this CAR T-cells and growing these up and bringing them back, and you can have an extremely effective immune response, but it's also a very strong immune response. So if there could be a lot of off-target toxicities, if the antigen, the target that you pick to activate the immune response to, is found on normal cells. Obviously, CD19 is on B cells, they're on normal B cells, and this therapy actually kills all your normal B cells. You can, though, as we've seen with patients, you can live without your B cells if you get immunoglobulin shots. So the problem with solid tumors, which I think Alex actually mentioned, is that we have not been really able to identify that single antigen that's really so cancer specific that we would be willing to use this CAR T-cell therapy to treat that cancer without having off-targeted adverse events. When it comes to the checkpoints, I think that the checkpoints have not been that successful yet with hematological malignancies. They have been so successful in solid tumors, and I think it really comes down to, obviously, with solid tumors, you do have to deal with stroma and the tumor microenvironment. Immunological malignancies, leukemia, the cells are usually, free floating in your blood system, so more amenable to be attacked by certain agents. But Alex, you may want to add response to that, since you're actually treating, someone that treats patients, then you might have a better answer than I do.
Yeah, I think again, solid tumors, the microenvironment is very complex, and I'm not to say that the liquid tumors are not, but I think as Jill mentioned in one of her diagrams that has 6 bubbles, it's not just about T cell activation, it's not about T cell killing tumors, but the T cell needs to get to the sites. And what we found, we've seen these examples in the past, with, for example, T cell therapy for melanoma, is that you can infuse a large number of antigen-specific, melanoma-specific T cells in the blood, and they can stay, up to 50% of your circulating T cells are melanoma-specific, but these T cells don't go to the correct site. And depending where your tumors are, in solid tumors, they might be at a site where either the vasculature is not hospitable, or the T cells don't have the right homing beacon to get there in terms of chemokine receptor. And once they get there, the different mechanisms that induces T cell dysfunction are very different, obviously, in different tumor microenvironment, different levels of hypoxia and different things. They all affect how effective the immune response is going to be. And that's why I said in my comment that the whole strategy in understanding tumor microenvironment, specifically for solid tumor, is going to be, I think, one of the next sort of big push to understand who are all the other players that really limit our ability to get a better response from the current and also additional future immunotherapeutics.
Thank you, Dr. Huang. Dr. Huang, you mentioned the challenges of long-term sequelae experienced by patients who are treated with the traditional therapeutic approaches. Is there any evidence to support the long-term safety of immunotherapies in children?
No, we don't. Well, so far, we don't yet have a long enough track record in terms of a specific, for example, checkpoint blockade. It's just not being conducted in pediatrics, so we don't know the answer to that. But we know, for example, patients actually survive bone marrow transplantation for salvage therapy or traditional as part of the mainstay therapy for leukemia, for example. They have suffered, the consequence can vary from some cardiac toxicity or endocrinopathy and to infertility and other things. And so we don't yet know what all the other potential long-term toxicities will be in immunotherapy. But I think given what we have seen on the adult side, I think there's enough, there's enthusiasm to allow us to maybe sort of imagine that there will be, potentially good outcome for patients undergoing immunotherapeutics. But I think these remains to be tested.
Thank you, Dr. Huang. This is another question you both may have responses to. I'll start with you, Dr. O'Donnell. Is there any evidence that patients can be primed for immunotherapies to be more effective?
Yeah, I mean, again, I think that we get this question a lot is, when you hear that your immune system can be used to treat cancer, can you do anything to bolster your immune response, as a preventative to get it? And I think there really is no proof of that. These are all manipulations of the immune system that are there, and I don't think that we really understand ways. I think I've heard many oncologists and clinicians and immunologists say that the best thing is to do what your mother said and, eat your vegetables and eat a balanced diet and have, but again, these are manipulating your immune system when a patient has cancer. This is not something that is being looked at in terms of as a preventative strategy in a normal population. Alex, I'm sure you can add something too.
Yeah, so I mean, I think one of the hypes about the HPV vaccine, right? So there are certainly instances where HPV vaccine being one of them, we know that at least the hope and the indications are that we can actually use the immune system to generate anti-HPV immune responses to prevent the development of HPV-related cancers, like cervical cancer and some type of oropharyngeal cancer, and that those are the type of preventive immunotherapeutic approaches that I can think of. But in terms of other cancers, it's a little difficult to think of right now. But HPV has been at least being one of the primary examples where you can actually use vaccination strategies to prevent HPV-related cancers.
Thank you both. This question is for you, Dr. O'Donnell. Your statement on the uniqueness of tumor neoantigens suggests that liquid biopsies may not be effective in detecting them. Do you have any thoughts on this?
Yeah, I think, the hope would be that perhaps there is a way from a liquid biopsy to identify in the blood, but I think we all know that not necessarily what's happening at the tumor site is actually the same as what's happening in the blood. That's certainly the case when you're measuring immune responses. Obviously, we have to, in looking for these neoantigens, it is really a genetic analysis. So a liquid biopsy that could pick out the cancer cells, which it obviously can, you could do a genetic analysis on that. I don't know that we're there yet, but I think that the future that may be potentially responsible, because it's basically predicting from the genetic, looking at the DNA in a cancer, predicting what would be an antigen that would develop that would be seen by the immune response. So you just really need to be able to compare normal and tumor cell genetics. And so I guess if you have a liquid biopsy that you can sample cancer cells that are representative of what the cancer cell does look like at the tumor site, that you would be able to do that.
Thank you, Dr. O'Donnell-Tormé. And unfortunately, we have time for just one more question, and this is another question that I will pose to both of you. We'll start with Dr. Huang first. Is it possible to identify patients who may respond poorly with adverse effects before they start treatment?
I think this is actually a very important question, and I think, and this certainly is an area of interest when I was discussing on the Moonshot, the President's Moonshot panel. I think we all want to get there. I think right now there are certain indications the patients that have severe toxicities also are the ones that tend to respond in terms of their tumor response to, for example, checkpoint blockade. But ultimately, whether we can do a better job at predicting who is going to respond to what agent and what approach and who is not, this is something that's ongoing right now. And I don't know if Jill might have a better sense where we are in terms of the CRI community in this front. I know there are many of the CRI investigators are involved in this whole effort.
Yeah, I think it is one of the major questions. Not only toxicity, not knowing who's going to have severe toxicity, but who's going to respond. And I think that is where the future is. And I think there is an awful lot of research, and I think we pointed out to some of them even like what immune profiling a patient to begin with, what their immune system looks like, what their microbiota looks like. There's a lot of different things that can affect, and I think we just don't know yet what it is. And it's only going to be through supporting research, right, from the basic right through the clinic to be able to answer that question. But I think certainly that's where the future of the field is moving.
Thank you both. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers. Their email addresses will be shown on the screen. As a reminder, the webinar will be archived on the scientist's website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. O'Donnell-Tormé and Dr. Huang, as well as our webinar sponsor, Beckman Coulter Life Sciences. Thank you, everyone, and goodbye.